We now switch gears only slightly. We stay in the area of peptides. And it's my very great pleasure to hand over the microphone to Sebastian Pomplun, and I hope he did not change the title of his presentation. Yeah.
I don't know what I said.
Okay. So, yeah, he's also from Leiden University, but from the Institute of Chemistry, like Sander before. And he will tell us today about synthetic transcription factors. And
yes,
very interesting topic.
So really happy to be here. And yeah, a little bit like hearing the talks like from yesterday and this morning. And I have the feeling that we are doing something completely different. Like it's about delivering stuff, delivering big stuff inside cells. But I will talk about a self-delivering protein therapeutic for nuclear application, which is like pretty cool because like it goes in by itself. It doesn't need any delivery system. So just to give a little bit of context here, I mean, this is a really simplified vision here of what was called the central dogma of life science, but we all know that it's much more complex. It's not just like DNA to RNA and then to proteins. There's a lot of stuff involved in all the system. Non-coding RNAs, transcription factors that decide which genes are transcribed when and how much and all this stuff. But clearly, this is still the central system, how our cells work, so any dysregulation here will lead to severe diseases. And I will talk about one particular transcription factor system, that is the transcription factor complex MIG-MAX, which is this, we don't have a pointer, it doesn't matter. So you have these two proteins that form a heterodimer and then bind to a specific DNA sequence, CAC-GTG, which is called enhancer box. And this activates with the transactivation domain here on the green part, which is MIG. It recruits cofactors and this activates enhanced gene transcription. And there's a natural counterpart because MIG cannot homodimerize, but you see that the blue part, which is MAX, doesn't have any transactivation domain, and MAX can homodimerize and then bind to the same DNA sequence. So now if you have the MAX-MAX complex, no transactivation domain, it will just block that enhancer box, while if MIG is there, it will activate gene transcription. So in a healthy cell, the system is kind of in a balance and everything is fine. But if you have MIG overexpression, then you will get overactivated transcription, and this enhancer box sits in front of a lot of gene programs related to proliferation, cell growth, and cell survival, all stuff that leads to cancer. So MIG overexpression is really not so good. Indeed, MIG is overexpressed in over 50% of all human cancers. So this is really the master regulator in all human cancers. So the problem is that now, if you see the structure, like we are not talking too much about medicine or chemistry, I guess, in this conference, but everybody will appreciate there is no binding pocket on this thing. So what you have is a coiled coil, seems like two sticks together, no binding pocket, and then you have a completely disordered transactivation domain. So there's no way to target MIG and MAX in a pharmacological way. But there's a natural product, actually, that's already on the slide that's targeting MIG-MAX, which is Max-MAX. It's not a small molecule, it's a pretty big natural product. It's a protein drug, no, that's blocking it. So the idea of this project started like, we say, like, can we make a synthetic protein complex mimicking this Max-MAX activity to block gene transcription by blocking the E-box side? And yes, I mean, then you have this not peptide, you have this protein. It's not even a protein, it's a protein complex. And then we need this protein complex to get inside the cell and get into the nucleus and then find the right DNA sequence and block it. So this is pretty challenging. But we were kind of hopeful because of two reasons. One, we were not the first people working on this. So there's one drug inspired by this thing, which is kind of an homologue of Max, which is doing the same thing. It's actually since 2021 in human clinical trials, because it kind of works. But we were more intrigued of making these stabilized complexes because this drug inspired by Max can bind to itself, can bind to MIG, so it's not really clear what's going on in the cell. So we really wanted to have the stabilized complex that can only be together and not then bind to the overexpressed MIG and block the DNA sequence. But the cool thing is that transcription factors bind to DNA, and the DNA binding region of transcription factors behaves like a cell-penetrating peptide because many times it's a polycationic alpha helical peptide. And from other studies in our lab, we had evidence that if you connect multiple cell-penetrating peptides in the same compound, it actually not only still works. It has a synergistic additive effect, so it gets even better. So in this compound, we would have 2 positively charged cell-penetrating domains driving the compound inside the cells by itself. Now, I mean, I will go really fast on the chemistry. I mean, for us, I don't know, I'm a chemist. This was the most exciting part. It was also a little bit playing around with chemistry and making these molecules, because it's important to appreciate here that the upper part where we want to place the linker to put these 2 things together are both C-termini of these proteins. So you cannot just like express this as a fusion protein, because you need to connect a C-term to a C-term. So you need to do some chemistry actually to put these things together. And so there are 2 ways of doing this. Like either you make 2 monomers and then you connect them with a bioconjugation strategy, or you synthesize this thing chemically in parallel on a linker, making 2 proteins in a row. So really quickly, like we talked a little bit about bioconjugation, so maybe I will stop just quickly here. So one of the things in bioconjugation is speed, and this is because like you cannot have super high concentration of these big molecules compared to a normal small molecule organic reaction. We are not doing it in vivo, but still we are dealing with things, and whoever is in the bioconjugation business might appreciate that the problem of kind of bioconjugation of a small molecule to a big molecule might have been solved, while the conjugation of 2 big molecules together is even more challenging, because now you have 2 partners that behave poorly in a chemical way and move slowly and have solvatation spheres, and you cannot like have them at high concentration. So we needed a really powerful reaction, and we used this palladium oxidative addition complexes. So we made these, so we decided to make these protein monomers with a single cysteine on the C-terminus that would react with this bifunctional reagent here in yellow, which on one side is already activated as a palladium oxidative addition complex, and on the other side it has still an aryl group, an iodo group. And the nice thing is, in the moment it reacts with the thiol group on the protein in a completely chemoselective way, the palladium is not kicked out, but translocates to the other side and creates a palladium oxidative addition complex on the protein with the bioconjugation handle, and this now can react with a second thing. And this reagent is super fast. It's much faster than maleimide conjugation. It's in the range of the tetrazine click reaction, so it's impressively fast. And you could use this, if you isolate this intermediate protein oxidative addition complex, you can make heterocomplexes, or you can just leave it in solution and then form the homodimers just in a one-pot fashion. So yeah, we made these proteins, and then we did this palladium catalyzed, not catalyzed, this palladium-mediated protein-protein conjugation, and made all the different possible homodimers of these proteins, MIG-MIG and MAX-MAX, and this analog that is in clinical trials, OMOMIC, but this was also just a little bit for the biophysical studies, how these different variants behave. And then we also isolated the intermediate protein palladium oxidative addition complexes, which is actually pretty cool that you have like a full protein with a palladium complex on it, and you still can react it like doing classical medicinal chemistry, but with really big molecules, because now you can put them all together in different ways. And we managed to get them all. And then really quickly, I mean, I really go fast here. We also were able on a synthesis machine at the time. I did this synthesis at MIT in the group of my postdoctoral supervisor, who has a flow peptide synthesis machine, and we just put a bifunctional linker and we synthesized these 2 proteins in parallel on this linker. So it's 90 amino acids plus 90 amino acids. So we did a synthesis of 100 amino acids in one shot. It took approximately 3 hours to do this. And then we got actually pretty decent materials out of these things. But yeah. I mean, it's pretty cool, but I don't have this machine, so I will not talk too much about it. Like one thing that's not really science-related that I just want to say here, that I did all this together with my friend Muhammad, who is now professor in Israel, and we're really thinking a lot about him, because he also just would like just to do science, but there's unfortunately a lot of other stuff going on there. Yeah, so we did all the biophysical characterization of these things we have made, so they're alpha helical, they behave well, they bind DNA, and so on and so on. But we wanted to ask the question, like, do they work actually in cells? No? So the first thing we did to check if they actually go inside, we put a fluorophore on them, and we did some microscopy and flow cytometry, and you can see that it first accumulates into the endosomes. It gets inside the cells, but it also over time accumulates into the nucleus. Actually, it takes a little bit longer. But then you still don't know. I mean, was the full thing cleaved and it's just a fluorophore going somewhere? Is it still functional? So the really important thing is actually to check if there's some activity. So MIG activates cell growth. So in MYC-dependent cell lines, you want to see that you get an inhibition of proliferation. So we did cell proliferation experiments in different cell lines with different levels of MYC, and we got really nice data. So in a low micromolar range for the most MYC-dependent cell lines, and then we get less activity the less MYC-dependent the cell line is. But there still could be some unspecific toxicity, so to be really sure that like the things that we see are actually what we want to see and not just like killing any type of cells and just some sensitivity limits, we did full RNA sequencing of these lung cancer cells. And then we got these gene expression profiles, and then we got really significant correlations to MYC-dependent genes that were up and down regulated. I mean, clearly you would expect down regulation, but all these effects are clearly really complicated. Something could be downregulated with upregulate something, so you see clearly also some upregulated genes, but we could call out a lot of these things that are clearly related to the MYC pathways, and also by gene set enrichment analysis, there were significant correlations of these genes. So we are really sure that our compounds get inside the cell, block EBOX-driven transcription. And just like in the last part, I mean, you saw these activities here. I mean, we had to use still around 10, 12 micromolar concentrations to get good data in these experiments, which is still a pretty high concentration. Now, if you correlate this, if you just do a binding affinity of this compound to DNA, the binding affinity is 20 nanomolar. And then the cell activity is 10 micromolar. It's like almost 1,000-fold more. It's kind of, there's a big mismatch. And the reason is still, we are getting inside cells, and it's really cool because it's like a protein complex therapeutic that's going inside cell, but probably still it's not really efficient. Maybe only 1% or 0.1% of the thing is getting in because it's still too big and still not well behaved enough. So what we are doing now, actually, we are investigating strategies to make this compound smaller by keeping the domain, which is both driving DNA binding and cell penetration, but removing all the other parts and replacing, at this point, with more chemical modifications. You clearly need more chemical modifications because you still need the perfect geometry of this complex in order to bind the correct DNA sequence and not just any kind of DNA. Transcription factors are pretty promiscuous. So we are trying in my lab to make different sizes of the thing, and actually there's pretty steep activity differences. So some of these compounds like do not show any activity, not even binding activity, and some of these compounds are really good. And I mean, I cannot show a structure here because like we did not patent anything and publish anything so far, but we are kind of close, so I just want to be safe here. And you never know who was in the audience, and Matthias already is like looking. But just what I wanted to say, like what I said before is, OmoMYC has a 20 nanomolar binding affinity and then 10 micromolar IC50 in cells. Now we have a compound that has around 100 nanomolar binding affinity to DNA, but in a reporter gene assay, it's 500 nanomolar. So it's actually really close to the binding affinity level, and it really shows that this compound is much more efficient in getting inside cells. And you can also see here on the left side, there's a dual reporter gene assay. So the reporter that's not MYC-related is completely stable, so it's not about unspecific toxicity here, but the MYC-related reporter goes down and has this really nice KD, and this is like, of all the published MYC inhibitors so far, this is the most powerful MYC inhibitor that I have ever heard of, so we are pretty confident that we can get a little bit further with this thing. Also knowing already that the parent compound OmoMYC is in clinical trials, so even if it seems a little bit interesting, like who knows how it behaves in vivo, but it seems to work. And with this enhanced version, we hope that we can get a little bit further in treating several forms of cancer. So with this, I thank my group at MIT and my group now, with whom we're working in, and our funding agencies, and you for your attention.
Thank you very much.